Hello and a very warm welcome to today's NTD Connect. I'm Marianne Compere. I'm speaking from the International Society for Neglected Tropical Diseases. Apologies, we kept you waiting for a few minutes. First of all, to build up the suspense and the anticipation for today's amazing talk. Also, just to make sure all the speakers were connected. Thank you so much and welcome to our audience. You're already very busy in the chat. It's really lovely to see everyone introducing themselves and joining in what promises to be a very interesting, very fruitful and enriching discussion today. Warm welcome, Akinola Oluwole from Sightsavers Nigeria, Annabella Chimpong from Ghana, also Joseph Vakrusi from Ghent University in Belgium. Really lovely to have you here today for what promises to be an important piece of work, a wrap-up of an incredible project for NTDs, but also for WASH and a better understanding of the role of precision mapping in understanding infections and particularly hotspots. We're here today. It's my pleasure to welcome our speakers one day before World Environment Day, who will be talking to us about precision mapping of human water contact and implications for WASH. Findings from the SchistoTrack cohort. On screen at the moment with our third speaker coming on and off, but hopefully, as always, I keep the faith that everything will absolutely fall into place. But on screen with me, please join me to welcome very warmly Associate Professor Juliet Chammy. Hello, Juliet. Hi, nice to have you here with us today. You are Robertson Fellow at the University of Oxford's Big Data Institute, and you're going to be introducing us to the SchistoTrack project, an incredible program, really, combining household surveys, direct water contact observations, GPS loggers worn by the study participants to get a better understanding of how water contact affects infection in the context of schistosomiasis. And we'll be also hearing how this understanding will enable better and more effective interventions in schistosomiasis control. Giving us the main part of the presentation will be Fabian Reitzug. Fabian, a huge welcome as well. You are with also with the Universities of Oxford's Big Data Institute, and you'll be taking us through the detail of precision mapping of human water contact. Welcome. And hopefully, technology being on our side, we will also hear from our third speaker, Dr. Narcisse Kabatarene, which I'm sure many of you know, from the Ministry of Health Uganda, from the Division of Vector-Borne Diseases and Neglected Tropical Diseases, to take us through a little bit of a better understanding of how precision mapping, how the output from this project can really influence on the ground the national NTD programs, but also WASH interventions. So a really packed session, really interesting angle on ecologically focal diseases, such as schistosomiasis. Without any further delay, just another very warm welcome to our speakers. Another big thank you to the audience. Please continue to introduce yourselves. It's lovely to see lots of familiar faces, names, sorry. And don't forget, whilst the presentation is going, if you have any questions, comments, maybe some findings or some experience from your own work, please do go ahead and use the chat to ask all those questions and to share any resources or any thoughts you may have. Well, enough from me, and it's my pleasure to hand over to you, Juliet. Welcome to all, and I hope you have an amazing session.
So I'm really happy to be here today to share one component of SchistoTrack's work. So we're going to be focusing on exposure, but the cohort is actually much broader than that. Schistosomiasis, as we know, is a global public health problem. The WHO estimates that 50 countries are endemic with schistosomiasis and need regular treatment, even though it's been reported in over 70 countries. And in these endemic areas, chronic cases are quite common, especially for the intestinal forms of the disease. Here, we focus on schistosoma mansoni, which is quite common in sub-Saharan Africa. And we think of chronicity in a number of ways. We're interested only really in chronic schistosomiasis. When I mention the word chronic, it's been used quite broadly, I'd say, within the literature, policy documents, but here I'm referring to two main aspects. One is the chronicity of the infection itself, because the flukes can live for quite a long time. There's a poor response to available praziquantel treatment, and most importantly, individuals are repeatedly exposed, and the treatment itself is not protective, so individuals are quickly reinfected. There's also chronic morbidity, which is what we're trying to divert from the eggs that actually don't get expelled within the stool. For the case of intestinal schistosomiasis, there's morbidities that are a bit more treatable than others, such as anemia versus untreatable morbidities like severe liver fibrosis. There's morbidities that are specific to schisto and more general to other infections or co-infections that occur in the area. And then you have morbidities of long duration. All of these aspects together are what define chronic schistosomiasis, and everything is reliant on repeated exposure. The SchistoTrack cohort itself is just trying to focus on these chronic elements, and we have three main goals. The first is to develop granular measures of current and past exposures, since we are following individuals over time. And the hope there is to understand the contribution of cumulative infection, residual infections after treatment, and chronic infections that don't resolve, and how they relate to morbidity. The main purpose of the cohort is looking at multimorbidity development. When I mention multimorbidity, I mean the existence of two or more chronic conditions. And here we're looking at multiple conditions within the liver and also co-infection interactions. So that our primary goal of achieving these two kind of sub-research aims is to develop tools for morbidity management that can be used within primary healthcare centers. SchistoTrack itself was developed out of collaboration with the Uganda Ministry of Health and the Division of Vector-Borne Diseases and Neglected Tropical Diseases. I myself have been working in Uganda now for about over 16 years, and this particular cohort started because of a number of studies led by me, Dr. Narcis Kabatrani, and Dr. Adrita Tukahebwa, who's not here on the call today, but many of you may know of her, where we were evaluating the effectiveness of mass drug administration and saw consistently that there's imperfect drug coverage and also a lot of residual morbidities remain. So there was a need to establish a cohort that looked at morbidity and repeatedly looked at measures of exposure. SchistoTrack itself is set up in 3 rural districts of Uganda on the east and western sides of the country. So these districts were chosen to represent very diverse populations, diverse tribes, diverse incomes, diverse access to healthcare, and most importantly, really diverse morbidity outcomes and exposure patterns. We are in Pokwach along the River Nile next to the Democratic Republic of Congo, and Bulisa next to Lake Albert, both of which have very highly mobile populations, and also in Mayugi district, closer to Kenya on Lake Victoria. SchistoTrack is running for, it's still ongoing, 5 years. It's a community-based cohort where everything's done at the point of care within the village. It's a prospective study, so we're looking at how exposure changes over time, but also trying to preemptively predict risk factors for morbidity. We have mainly an annual follow-up, and everyone within the cohort is treated with routine praziquantel treatment, and there's a drug efficacy point to look at clearance measurements and also seasonal time points to look at variations in infection. At the moment, we are in 52 villages. We've recruited nearly 2,200 households. These households are randomly sampled from village registers so that we can get a population level representation of these villages. And then every household that we sample, we collect information on every single person within that home, mainly socio-demographic information and medical history information. And those individuals are aged 1 year and older. And then for the clinical sub-study where we're collecting samples and we're looking at infections and conducting clinical examinations for different morbidity outcomes, we take one child and one adult from each home so that we can look at generational correlations as well. And at recruitment, a child is between 5 to 17 years. We had a 5-year limit because at that time, those were the individuals eligible for praziquantel treatment and older, and adults are 18 years and older. So we have 2 people per home, and at the moment, over 4,300 individuals within the cohort. This is an absolutely huge effort. I always say that our teams are more than a village. We're a huge group between Oxford and Uganda with multidisciplinary expertise, combining quantitative methods with empirical approaches. And the key individuals within our teams include malacologists looking at the snails, surveyors looking at medical histories and sociodemographics, technicians looking at all the infection diagnostics of the individuals, HIV counselors getting detailed histories on that and the co-infections, nurses conducting abdominal palpations and other routine anthropometry, and sonographers looking at spleen and liver measurements. So although our key outcome is liver fibrosis, we look at a number of morbidities related to that or different causes that could contribute to liver fibrosis. Our key infections are not only schistosomiasis, but also malaria, hepatitis B, and HIV. And a number of our outcomes that we're looking at are reliant on assessment through either ultrasound or peripheral blood. But what we're focusing on today is actually the really diverse multimodal data that's collected to understand human exposure. So here, specifically referring to water contact. Within the SchistoTrack cohort, we have conducted household surveys, as I mentioned, with everyone within the home, age 1 year and older. We have very detailed water, sanitation, and hygiene measurements. And we've adapted measures from UNICEF and the WHO to try to maintain some sort of international standard and consistency to all these surveys. We have direct water contact observations where individuals would sit by the shoreline and record the degree of bodily immersion, contacts of individuals over set periods of time, about 2 weeks. And today, which will be a key focus of the presentation from Fabian, we've given out wearable GPS loggers and tracked individuals over 2 weeks, both children and adults. We also collect all the household locations, so we know the distance of all the homes to the water bodies. But in addition to that, we've done full infrastructure mapping where we visited every single tap and borehole and public latrine that are within our study villages and collected the GPS points of that, as well as all the private drug shops, health centers, and schools. So we really have incredibly rich data to give a more granular perspective on human water contact. And before I hand over to Fabian, just the absolute bare minimum of statistics to understand kind of what this cohort looks like. So we've had a couple of expansions. So here I'm just showing the baseline recruits. For individuals within all the households, age 1 year and older, we have an even distribution of males and females. It's quite a young population with the median age of being 18. But something I want to point out here is that we've collected information on every single person within these homes. It is a rather young population where it looks like there might be some out-migration of the teenagers. And then for schistosomiasis prevalence, the minimum prevalence as assessed by microscopy is over 43% across all of these districts. And then if we looked at lateral flow test using point of care circulating cathodic antigens, and we treated the trace results of those tests as positive, then the prevalence would increase to up to 66% as a starting point. So really, these are highly endemic areas. So I hope that gives you a quick overview of SchistoTrack, and I'll let Fabian take over to show really the exciting results of what's ongoing with our analyses related to human exposure.
Yeah, thank you so much, Juliet, for introducing the study. And I also want to start off with two things. First of all, I want to say that a lot of this work has been part of my PhD that I just completed at the University of Oxford that was supervised by Juliet. And I also really want to give another shout out, especially to our Ugandan team at the Vector Control Division, because I mean, their efforts have been really, really instrumental for making this happen. I mean, Dr. Nasis Kabatarena will speak after me, who has been spearheading these efforts on the Ugandan side, and also Betty Nabat, our field manager, has been really crucial for making this very extensive field work happen that was instrumental for a lot of the findings that I will be presenting. And so now let me delve into the topic of precision mapping of human water contact and the implications for WASH. And I want to start out broadly by saying that, first of all, I think we can all agree that there's a lot of common ground between WASH and NTD collaborations, because we have these common shared goals that WASH and NTDs are addressing. Because if we do WASH right, I think that, and that's also kind of what the WHO guidance on this topic shows, is that there is a lot of room to tackle like a lot of different NTDs in one way or another. But the argument that I'm trying to push a little bit in this presentation is to say that I think that like the crucial missing piece here that we kind of need to better understand is human behavior. Because, I mean, the WASH is kind of building on this assumed theory of change, where we have the idea that, okay, if we implement WASH interventions, we're somehow going to change human behavior, and that it's going to reduce the burden of infection for the diseases that we care about. But I would argue that currently we do have somewhat limited understanding of some of these intermediate behavioral outcomes that we're trying to assess. And I think that we also need to be like quite disease specific, because like the at-risk behaviors that we care about, they depend on which NTD we're actually talking about. And I think once we understand that for kind of like these singular diseases in the first instance, I think we can have more of a bottom-up approach of then looking where the synergies are across NTDs to kind of get towards or move towards a broad WASH and NTD collaboration. And so next, I mean, I kind of want to say that the singular disease that we're focusing on, as you may have gathered, is, of course, schistosomiasis. And I think a lot of you are familiar with schistosomiasis, but just to bring everybody onto the same page, I am showing the life cycle here. And the most important part that we're obviously focusing on is human water contact. And this is when people come in contact with the parasite. So the parasite is waterborne. It burrows through people's skin, gets into the human body, it matures in the human body. Then the eggs that kind of result from the pairing of the parasite, they are then shed via the feces or the urine, depending on the species, and then they pass through freshwater snails as intermediate hosts, and then from those snails emerges the infective stage that can infect another human being. And what has made the control of schistosomiasis so challenging is that we have a lot of heterogeneity in infection prevalence on small spatial scales. Or said differently, that in one village we can have very high prevalence, in the next village we have like a much lower prevalence, and it is still poorly understood why exactly this is happening. And because of this heterogeneity, people have focused on this concept of precision mapping of infection prevalence, right? Where we say that, okay, we're dividing up a district into smaller units, such as, for example, like the sub-districts, and even smaller units like so-called transmission units, where, for example, this could be a school, and we're measuring the prevalence in each of these units, and then we're looking at how we have different prevalences in different places, and we could then use that to target treatment, for example, to these different units based on their prevalence. But I think it's well understood now that, I mean, treatment alone is not going to be sufficient to interrupt transmission, and that we need to think more broadly about like multifaceted strategies. And for those strategies, we need to understand not only what is the prevalence, but also like what is actually driving the prevalence. Like, why do we have high prevalence in like this one transmission unit, but not in this other one? And what is even the size of these transmission units that is appropriate here? And that's why I would argue that we also need to have precision mapping of water contact, because this is what could explain why we see these differences in prevalence that are well documented. And when we think about water contact, right, we really need to understand, okay, like what are the kinds of occupations that people have, like how much access do they have to safe water? We also care about human mobility, environmental factors because they're driving water contact and environmental risk. And so I think that we kind of need to think about how we can integrate precision mapping of water contact with the precision mapping of infection. Now, I would say that like not so much work has been done around precision mapping of water contact. And the reason, of course, is that water contact is quite complicated and it's probably resource intensive to measure water contact. But what I want to convince you of in this talk is that, first of all, we should care about water contact. I mean, even if we only care about mass drug administration, but we especially need to care about it if we care about multifaceted control. And I will also argue that there are actually ways in which we can measure water contact in ways that I think would be relatively cost effective and feasible, even within like control programs that don't do a huge amount of data collection. And so now let me kind of dive right into the talk. And this is kind of starting off a little bit with like where my PhD began. And that was like a very simple question that I tried to answer, which is around like just how is water contact actually related to infection? And that may seem an obvious question, but of course it is true that, I mean, when somebody's infected, they must have had water contact at some point, but it doesn't mean that they're having a lot of water contact like now. So we kind of need to understand to which extent we can actually kind of equate infection to current water contact. And to investigate that question, I did a systematic review. I identified roughly 100 studies that were reporting on associations between water contact and infection. And I found that there is a relationship between water contact and infection. No great surprise here. So we found that reporting any current water contact, and that is mostly based on self-reports, we found that self-reported water contact is basically associated with 3 times higher odds of infection. And we also found that there were, however, like a lot of factors that influenced basically how risky water contact was. So you can see here on the top, for example, like the endemicity setting, that villages that had a higher prevalence, in those villages, having water contact was more strongly associated with infection, presumably due to the fact that there's probably more like a higher environmental risk present. So the same water contact confers a higher risk of infection in those settings. And then we also found differences by the types of water bodies, the nature of the water body, not water flow, but also importantly, for example, based on climate. And there we showed that especially tropical savanna climate, and that's like climate that has a very pronounced dry season, was kind of associated with higher risk, showing that, seasonality is also an important factor that matters for how risky water contact is, because we know very well that within the dry season, people are often like kind of crowding around those remaining water sources that haven't dried up and that could be associated with a higher risk of infection. But there were also important gaps that were still not answered by the systematic review. And the ones that I want to point out here is that most notably, even though a lot of studies talk about it, we weren't able to understand what is the variations by age and gender, for example, in these water contact patterns and the associations between infections. And that then really opened up kind of a whole new avenue of research that we started with the SchistoTrack data to better understand these water contact patterns and ultimately the implications of these different water contact patterns for control interventions. And so I kind of want to just give a little bit more of a feel for like what is the setting that we're actually operating in with the SchistoTrack study. I mean, Juliette has already started to give you some details, but here you can see a typical water site. This is a photo that I took where you can see, children and women getting drinking water in these yellow jerry cans. And you can see there's also animals present here at that water site. And quite importantly, you can also see that vegetation here that is partially submerged, and that is the habitat for the intermediate snails. So we can really expect that this site is quite risky and that people are probably getting infected at this site. Just to give you some more statistics around the setting, it's like we're really focusing on very high endemicity communities within 2 kilometers of water bodies. They've had like 13 or more rounds of mass drug administration with praziquantel. The infection prevalence is above 40%. We have very high levels of self-reported water contact, nearly 50%. And people live quite close to water sites, but also to taps and boreholes. And that will become quite important, I think, in the later part of the talk. And so the first question that I already alluded to is we wanted to understand, okay, how do these water contact patterns actually vary over age and gender? And the interest in understanding that is that it is kind of a core assumption, for example, in mathematical transmission models, that the levels of water contact over age are somewhat proportional to the levels of infection over age. And that is something that we wanted to investigate. And it's also often been said that males have higher levels of infection because they have higher levels of water contact. And we also wanted to see if that is actually true. So when we investigated just descriptively, what are the patterns of water contact and infection over age, we started to see something that we found rather surprising. So here in this graph, you can see the black line showing the infection prevalence over age. And this follows kind of this typical shape where you have a peak of infection around the ages 10 to 15, right? Where you can see that almost 70% of children in that age group are infected. And then it kind of declines as people move into adulthood. But what we saw when we looked at the water contact patterns, we actually saw that the water contact patterns are actually comparatively low in these children. And the peak of water contact in across all of our different study districts in Uganda occurred much later in life, around age 30 to 35. And so this already tells us something interesting here, saying that we can't really use infection always in all contexts as a simple approximation of the levels of water contact that people have. And we also see that children are getting infected despite having comparatively low levels of water contact, and that furthermore, like the infection prevalence then, as people like move into adulthood, it declines despite levels of water contact, or the proportion of people with water contact increasing, which is somewhat intriguing and possibly explained by dynamics of acquired immunity. But I think that you may be wondering, like, okay, like now we're just showing kind of like overall water contact, but we haven't really shown, like, for example, what is the impact of duration and so on. So we also investigated like those patterns here, and also looked especially at those gender differences. And here we also saw that it's a little bit more complicated, because like whether males or females have higher water contact depends really on what metric you're looking at. Because on the left-hand side, you can see that females have a higher frequency of water contact, right, as you can see. But then when we look at the right-hand side, we can see that the duration measured in hours per week is higher for men than for females. And of course, the likely explanation for this is that males and females likely do very different types of activities, and they have different frequencies and duration, and that explains what we're seeing. And this can be shown really like with these next visualizations that we looked, or that we created to look into this, right? Where you can see here that, for example, for children, so if we just focus on kind of like the very left-hand side of the graph, right, we see that there's a lot of blue, which means that for male and female children, like domestic water contact activities, such as like doing laundry or getting drinking water, that's what dominated. But then when they moved into adulthood, we see that those patterns like change, because in panel A, you can see how that beige color becomes very dominant for males. So that means that they then have mostly occupational water contact. So for example, fishing or agriculture, while in the bottom panel, you can see that for females, domestic water contact remains the major source of their exposure. And I also just want to show that this basically remains the same when you also look at the frequency and the duration of these activities. So the next step then is to also think about, okay, like what does this mean? Because obviously the relevance of these different activities and understanding these patterns is that different activities may carry a different infection risk. And which activities are most risky, I think, is still poorly understood. I mean, here you can see one example from the literature where the authors said that laundry, bathing, and recreational swimming are among the activities that are the most risky, and the collection of drinking water may be unimportant. And we wanted to understand that better, again, using the same systematic review from before, where we basically profiled these activities. And to just back up a little bit for people who may not be so familiar with kind of like, okay, why do these different activities matter? I just want to say that, I mean, of course, different activities have different durations of water contact, different frequencies of water contact. They have different degrees of bodily immersion. So, if you're swimming, your whole body is immersed versus, if you're just getting drinking water, fewer parts of your body are immersed. And then also, of course, the timing and the season of day matters, because that impacts like how many cercaria are like present at a given point in time. And so we really want to understand like which activities are the most high risk. And what we found is that the picture is actually not so clear. So here we find that basically all of these different activities are associated with infection risk, right? So we see that even these kind of relatively short activities like crossing or urination or getting drinking water are still significantly associated with infection. Then when we group them more into categories, right, we can see that like these that occupational activities are slightly more strongly associated with infection compared to recreational and domestic activities. So there's some signal there, but I think overall, like from that review alone, I think that we still don't have enough grounds to say, okay, like this water contact is unimportant and this water contact is important. So I think that we have a strong argument to say we really need to think about basically all sorts of different water contact events that people have and think about how we can address those. But I don't think we can like leave aside like any single activity or type of activity just yet. And so the next part of the talk then also focuses like more around like the spatial dimension. And I think that's a dimension that is also still relatively poorly understood. And it kind of goes back a little bit to that precision mapping of infection prevalence, because we know that for infection, we observe these gradients, right, where people have said, okay, like, you know, let's focus treatment on, say, for example, 5 kilometers around water bodies. You know, some people have also shown that, you know, people still 10 kilometers away have like higher infection prevalence. And we also need to understand like how is this actually looking when we look at water contact? How do these gradients look for water contact? And also at what level should we measure these gradients, right? Should we focus on like household gradients, village gradients, or school gradients? And just to, you know, say like what a gradient is, it's basically kind of like what is that relationship between distance and levels of water contact or levels of infection, if you care about infection gradients. And when we looked at those gradients within the SchistoTrack study, again, we found something that may be a bit surprising, and that is that actually those gradients did look different for infection and for water contact. So when you look at the infection gradient here, you can see the red line. So this shows, okay, what is the prevalence of infection over household distance from water bodies? You can see that even people who live 2 kilometers from water bodies, they still had pretty high levels of infection, like around 40% still, right? But when you look at water contact, it actually looks different, because you see people who live right next to water bodies, their prevalence of water contact is roughly 50%, right? And then once we move 2 kilometers away from water bodies, only 10% of people still report that they have any water contact. And that really shows us that, again, we can't really equate these gradients of infection and of water contact necessarily. And I also really want to emphasize a little bit more that when we're looking at the SchistoTrack study, right, water contact was really very, very concentrated around the shoreline, because we see that 80% of all the people that self-reported water contact, they lived within roughly 400 meters of water bodies. So really very, very focal here. And then the next step was also to understand, okay, like now at which level should we actually measure those gradients? Because what you see here, this is basically measured at the household level. And we then also did some analysis where we basically simulated how it would look if we now had those gradients at the village level or at the school level. And what we see here is that for the village level, here in the middle plot, you see the trends look relatively similar to what we would have observed at the household. But then for the school, you can see in the bottom, like it looks actually quite different, telling us that simple kind of school distance gradient to water bodies does not necessarily approximate the true household gradient that we observe across water contact. And so this will require some critical thinking if we want to have both mapping of water contact and of infection at the school level. We kind of need to think about how that can be done. So then, and I think this is really some of the most exciting work that we've been doing as part of SchistoTrack, is to use these wearable GPS loggers that Juliette has already alluded to understand like basically people's movement patterns and how those patterns are aligned also with their contact patterns of water sites and taps and boreholes. Because a lot of people assume that people like mostly visit the water site that is closest to them. And with survey data alone, it's quite difficult to understand which water sites people are actually using, because it's really hard to describe like, oh, did I visit this site or did I visit that site and so on? And like, what are the distances to all of those sites? This is why the GPS data is really useful. And second of all, the GPS data is also useful because it gives us an understanding of like mobility. So we can also see not just water contact, but like just how far do people move in general. And to give you like some understanding of like how this actually looks like, I'm showing here like a little visualization from one of our districts. So we gave 400 loggers out to individuals as part of the SchistoTrack study. And here you can see in one district, I put a box around those 4 study villages, which and the GPS points are kind of like the yellow dots. So you can see here how people mostly stay kind of within those study villages, but some people go far. You can see those traces that go onto the lake, right? This is fishing activities. And you can also see kind of like this one yellow line branching out there more towards the north. That is people traveling along a road to a different town to, go to a market, to visit friends, to do other activities as well. And some people even go into the Democratic Republic of Congo here, as you can see. And so we are really interested in understanding, okay, like how far do people actually travel to do these different kinds of things, right? And we see that, when we think about visiting taps and boreholes, that is really quite local, right? Within the study, we didn't see anybody who went further than 1 kilometer to go to a tap and a borehole. Then when we looked at water sites, we didn't see anybody who went further than 10 kilometers to visit the water site, right? But then when we look at like how far did people move for any purpose, right? We see people do actually move kind of far, right? We see that people moved up to 100 kilometers for like different purposes. For example, like going to the capital city to, do business, to meet friends and so on. So like there are quite clear differences in kind of the overall movement patterns. And when we think about like the movement that was just related to kind of water contact, safe water contact and unsafe water contact. And this is kind of where we also started to think about more closely around like, okay, how are these movement patterns also different for different groups? Because people have said, for example, that, fishermen, like they're maybe more mobile than other people. And kind of this whole idea about mobility has been quite influential, especially around also like treatment for these groups, because when they're more mobile, they're more often missed by treatment campaigns and so on. And so we wanted to understand, okay, how far do these people actually move? And what we saw was that these patterns are also a bit more focal than people generally maybe thought. So we see that people who are fishing, who have a fishing occupation, so fishermen, like didn't really have different or a larger mobility than like overall adults, right? We can see that it's all kind of like in the range of 650 meters on average that they were away from their household location. But we do see one clear difference where we actually see, okay, adults have like a larger mobility range than children. And so that means that for children, kind of the number of water sites that will be available to them is like going to be smaller because they don't move as much as adults, which is, I think, something that people would have probably expected. And so now it's kind of like a little bit about bringing it together, right? I mean, I really want to hash out, like now that we've learned a lot about water contact, you may be asking, okay, like, so why should we care about water contact? And I think I want to make the argument that it really does matter for a lot of different kinds of interventions, not only behavior change interventions, but also other ones. And to illustrate that, I want to orient the discussion like around these 3 different buckets, which I think are useful for thinking about how we can intervene, right? We can intervene at the water site, so we can like reduce the environmental risk at the water site. We can build wash infrastructure, right? And we can focus on people by changing their behavior, like through behavior change campaigns and like shifting maybe behavior to like less risky times of the day and so on. And I want to illustrate how a better understanding of water contact can help us to make all of these different kinds of interventions that we can be thinking about like more effective. And first, I want to focus like on kind of the people and their water contact behavior. And so what we did, again, here with the GPS loggers is we tried to develop like some very simple models where we try to predict how much water site usage do we actually get depending on how far away these different water sites are away from the household. And what we observed is that there's really like quite a steep decay where, when a water site is just 50 meters away from a household, we predict that roughly 60% of people are actually using this water site. But then, once the site is like 250 meters away, it's only 35% of people who are using it. And then once we are like at 1,000 meters, right, only less than 5% of people are using this site, right? And I think that the really important message here is to say that, okay, like water contact is actually quite local. And I think that this is an important message because it says like, okay, if we care about water contact, we don't need to care about water sites that are like 5 kilometers away, 10 kilometers away. In these high endemicity settings, it's really the local water contact that matters. And in addition to that, we also see that like people actually don't visit that many different sites. So over the 10 days, we observed that people on average visited just like 1 to 2 different sites, but like not much more. So it's not true that people are visiting like 10, 20 different sites, right? But water contact is local with a limited number of sites. And I think that makes kind of the problem like maybe a bit more tractable because we can say, okay, like let's just focus on local water contact. How can we address this local water contact that occurs within the village or in the neighboring village? But that's kind of like the spatial scale that is mostly relevant here according to these analyses. And importantly, I think the same thing is true actually when we think about like safe water infrastructure, because what we see here with this red line is like we applied the same modeling approach to also understanding, okay, which taps and boreholes are people using? And we see that because that red line is below the green line, the blue line, we see that basically people travel even less to visit these taps and boreholes than these water sites, right? So we see that, okay, already when the sites are the taps and the boreholes are 250 meters away from people's households, right, only 20% are still using these taps and boreholes. And that really tells us that also when we think about safe water infrastructure, like it's really about bringing this infrastructure in the proximity of households, because if you build a tap or a borehole like at 2 kilometers from somebody's household, it's really very unlikely that they're going to be using it, or if they use it, they may be rarely using it. So I think that's quite important to understand that both of these patterns are very local. Then I think that these same kinds of models also have very interesting implications as well, not only for water contact, but even if we think about environmental control, and people have focused a lot on like snail control, also known as mollusciciding. And what we can see is that because we can pretty accurately predict like how many people are going to be using a given water site, we can use this information to then identify which water sites are the ones that are most used by the community. And that can help us to then target mollusciciding at the sites which are going to be most used at the community, because this is where people are probably going to get infected. And what I'm showing here is that we had predictions at the population level, and we got the locations of all of those households, even the ones that were not in the study, we got them from satellite imagery. And then we predicted for each of these households, like which sites are they probably going to use. And then based on that, we can get an estimate of how many people are probably going to use these different water sites. And there are some interesting patterns here, because you can see that, for example, there are 2 different sites which are very close to each other, but there's like very different numbers of people using them. So this one site is probably only used by, say, like 50 people. And then there's another site that may be used by up to 400 people, right? So you can see these very big differences. And those sites are probably just 100 meters away from each other. And so this tells us that, we can really identify like probably the most used sites, and we can use them for more targeted environmental control strategies if we want to implement those, showing that water contact behavior is also relevant for understanding how we can most effectively deploy environmental control. And then the last thing that I really want to focus on is thinking about the infrastructure, right? And this is really, I think, that something that's talked about a lot. And I think that water contact is relevant here because we can really try to understand to which extent do we actually expect this infrastructure to reduce open water contact? And we studied this with the GPS loggers, which I think gives an unprecedented insight into, how people are using water sites, how people are using taps, boreholes, and how these 2 things may be interconnected. And I just want to recap this basic theory of WASH that is kind of implicit here, which is that, okay, if we have a household that has high levels of water contact, right? I mean, the idea is that, okay, now we're going to build another tap and borehole close to that household. So the household needs to travel less far to visit this tap and borehole, and therefore that household will reduce their open water contact. And we wanted to understand if that is happening, to which extent that is happening using that GPS data. And first, I just want to give you like some descriptive statistics on that as well. So we see that actually the overall water contact duration is maybe not as high as you think, but it's kind of similar to what other people have observed in other studies, for example, in Senegal, where we see, okay, on average, people had water contact for like 3 minutes per day. And when we look at only the people that had water contact, I mean, their duration was roughly like 9 minutes per day. And I should also say that approximately 30% of people were using taps and boreholes. And these patterns were pretty similar across different groups. So, I mean, both adults, children, males and females, like kind of used taps and boreholes to a similar extent. So no very, very clear patterns here. And what we did is like those models that I was showing you, like those spatial decay models where we saw those declines, right? We basically built an integrated model where we said, okay, like, let's assume that these taps and boreholes like reduce somebody's open water contact. Let's quantify like how much they probably reduce people's water contact. And what we came up with is that taps and boreholes may actually reduce the duration of water contact in our study population by 31% compared to if there were no taps and boreholes present in that study area. And we find especially that fishermen, fishmongers, and people who are engaged in agriculture, they have still the most water contact in the study, which makes sense because these water contacts are not really impacted by having access to safe water. Because, I mean, obviously, if you go fishing, right, you will even go fishing if you have safe drinking water, right, if that's your occupation. But one important thing, which kind of aligns with what I said earlier, is and that's really a point that I want to stress, is that, again, we kind of really see the importance of focusing wash infrastructure on areas that are very close to the shoreline, because we estimate that, you can reduce maybe the duration of water contact by approximately 40% with safe water infrastructure if you focus it on areas that are like within 100 meters of the shoreline. And then like once you build it at like 1 kilometer, you have like hardly any impact, right? So really the high impact areas are those that are like within 500 meters roughly from the shoreline. And so that's really something important that we need to focus on. Obviously also being mindful of other like hydrological constraints and so on, because obviously you don't want to build taps and boreholes too close to the shoreline as well in case there is flooding and so on. But generally the impact will be highest relatively close to the shoreline. And the part that I want to end on is also just kind of thinking around like water contact measurement, because obviously the things that we've done around SchistoTrack is quite an intensive research study, as Juliette has also highlighted. So obviously the ways in which we measure water contact are far beyond something that like an operational program would do. But I do think that this study does hold some lessons for how water contact measurement can be done in operational programs. Because I think we showed quite effectively that actually water contact surveys are a pretty good tool to get an understanding of people's water contact patterns. Because if we focus on these key kinds of activities, domestic activities, recreational activities, and occupational activities, we are capturing like essentially all the different water contacts that occur, and we have these clear patterns over age, right, that we can capture. And if we also focus then in addition also on gender, age, and occupation, like those are also really important risk factors for water contact. So with a few survey questions, capturing those kinds of things, I feel like that we could have a good handle on kind of like mapping human water contact in kind of operational studies. And then I think that despite what you may think around wearable GPS loggers being very expensive and so on, I do think that we show that you could actually get away with just doing a relatively small scale water contact studies to understand like these gradients that we show in water contact. Because we estimate that even though our study had like roughly 450 participants, that like we would have been able to understand the general spatial gradient of water contact if we had just collected data from 10 to 15 participants. So one could imagine just doing a small scale study where you give these GPS loggers to 10 to 15 people, you map where all the water sites are, and you're able to understand the spatial decay of water contact. And doing those studies across different contexts could understand us, or could help us to develop more generalizable models of water contact across different contexts, and understand, okay, are those patterns actually similar in different regions, different countries, different continents, or to which extent are they kind of location specific? And so I want to just flag, that there is obviously a need for future work to assess to which extent, the things that we found generalize to other contexts, and to kind of collate more data to understand those gradients better, and to also experiment with these water contact surveys as part of operational programs. And lastly, I also really want to emphasize that while a lot of our work that we're showing here is highly quantitative, I mean, obviously there's also a huge role for qualitative work to understand, people's perceptions, what drives their usage patterns, which is kind of complementary and will help to inform future quantitative work as well. And with this, I really want to hand it over to Narcis Kabatarena now, who will talk about some of the policy implications, drawing on his very rich experience at the Vector Control Division of the Ministry of Health. So Narcis, I'll hand it over to you, and I'll switch to the next slide.
Okay, I'm Narcis Kabatarena from Vector Control Division, and I've been working on schistosomiasis for quite a long time. And from these studies, I could see that there is something we can pick from the study results, so that it can impact on our policy. Policies to control. Due to that, we thought that we could go to school, do prevalence studies, as well as do water contact studies. But we just realized that the prevalence studies have to be separate from the water contact studies, because the school is not a suitable environment to study water contact. Water contact studies should be done in household level, and as the speaker said earlier, noting that you have to look at the distance to know the distance from the water contact levels. Homes which are very close to the water contact, to the water contact, that's where actually you can do water contact studies in that environment rather than in the schools. We also saw that water contact we saw that there was high prevalence in it, while the prevalence itself is maximizing at 15. It just shows that the children have less water contact than adults in this setting, yet they are more affected. That implies that the reinfection is higher in children, much higher in children than in adults, almost twice as high. And that means that when we are limited with resources, it is very important at least to treat children, maybe treat them every 6 months, while adults, at least you treat them yearly. So in that case, you can save resources, you can save money, because you would not be going house to house to look for children. You can just see them in schools and so on, and give them drugs and treat them twice. But once you have to give to adults as well, you have to go to house. And we have seen that almost all water contact levels, all water contact types are a problem. You cannot know which water contact is safe. All of them have a risk. So if we have to use wash, and you hope that you will control the disease, you have to look at things which will control the disease, both for children, for adults, for women, but it is not like that. The females normally, as he said, go to water due to domestic issues. Children are in the water due to domestic issues as well as due to recreational. And the adults are in water mainly due to occupational. So if you provide fresh water, for example, safe water to a village, don't you expect that you will control water contact for males? No, males, because they go there for not safe water, but they go there because they have occupation to do. So you will not control their contact, and that means it will continue just as it is. Children who go for recreation will also not be saved. So once you put water contact, yeah, so we need to address when we we want to do wash, to use wash to control the disease. We need to consider all sections of water contact, but since it is difficult to have, let's say, recreational, you have safe water, you have everything, maybe when don't expect too much. You know that there are other sections for recreational which are going to the water. There are other sections for occupation who are going to the water. You have only saved the water, the fresh safe water supply will only save the children who go and the people who go for domestic purposes. And when you are giving health education, maybe you can try to pull others to the safe water, but it is not easy. So if you have to do it, it is better you put recreational areas for children as well, if it is possible. And you also consider protective gear for, let's say, adults and so on. Safe water, also once you put it there, he has shown you that people do not walk for 1 kilometer to go to collect safe water. If you put safe water, you look at the distance. It is those people who are close to the borehole that will utilize it. And the people, because they like safe water, they will also get closer to move to collect the water, but hardly more than 1 kilometer. If it is 1 kilometer away, they will just go revert to the natural water, and they will not go for that safe water. So where you will put it is very important. You concentrate at least 1 kilometer. After every 1 kilometer, you put their water. But above all, this study, what helps us, we have seen that on the lakes, it is not easy to control the snails. And moraciding kills fish and so on. But you can do focal moraciding, and you control the fish level, because some areas they will have refugee places where they can survive. You do it intermittently from place to you skip others and you go to. So you concentrate on those areas which have high water contact. And that's where we would do our snail control, either for environmental change by removing the vegetation which the snails feed on, or the things which protect them from winds and the waves, or by moraciding itself. But we concentrate on only parts which are having a lot of water contact. So water contact should assist us to identify those areas where the people, majority of the people, are getting into contact with water. Collecting water, what I have seen is that age, gender, occupation, they are very, very, very vital in the water contact. You don't have to study other things. I think these ones give you everything, and you just concentrate on them, and you can scale them up to other areas, and you still get the same results, because we move from village to village and from lake to lake, and we'll get to the same almost similar results. So age, gender, occupation are very important areas for data collection. Thank you.
Thank you so much, Gnosis. And a very big thank you as well to Betty Nabate, project coordinator of SchistoTrack. on the ground in Uganda. So hello, Betty. We can't see you on screen.
Hello.
Hi.
Hi.
Thank you so much to both of you. And Juliet, thank you as well for your really important and useful introduction. Fabian, that was an amazing presentation right off the back of your PhD Viva. We all are rooting for very good news as soon as possible. But all in congratulations really to all the speakers and the many colleagues, many I'm sure are on this call, who've contributed to this hugely collaborative project with such diverse data. And it's been a truly fascinating tour of this project. Particularly interesting in terms of shedding light on that black box of human behavior. So on this occasion, through the lens of human water contact and schisto. So honestly, it's been really interesting to hear how this ambitious work is coming together. And we can already see many, many applications for NTDs in so many different ways. So thank you. While you were speaking, a few questions have come up in the chat. So I might, in the interest of time, turn to those straight away. A very big thank you. And before we pull those up, there's been quite a few really warm congratulations. Narayana Sami, Guru Sami says, good presentation, informative also, congratulations. Amina Yusuf, thank you so much for very good presentations. And yeah, just all round, big round of applause all round. But just delving now into the questions, the first few that came up were just really about some of the data you presented, Fabian. Kalkiran Bagasho is asking about including the duration of contact, which I think you then went on to cover. And if there's anything anyone in the audience would like to revisit, I just wanted to remind you that this webinar is recorded and will be available hopefully later today. But maybe you could shed a bit more light. Here a question from Irene Larby. Did you collect any data on the specific times of water contact? Was that important or relevant? And also a question from Anna Kildemos. Any relation between the intensity of infection and the distance of the household to water bodies? If that is put into the equation, does the pattern or curve then resemble the water contact more?
No, thanks. I mean, those are two like very relevant questions, and I mean, let me start with the time question. So yes, we did actually collect also detailed data on the time of the day. Like, I mean, both. I mean, as part of the survey, and then also with the GPS loggers, like they also have a timestamp, so we know when people are going to the water. For the time of the day, I mean, like those patterns, we also found that they're really also specific to the specific district. So I think that, for example, in one of our districts where I also showed the GPS loggers Pakwatch, we saw that there, like a lot of water contact was concentrated more like midday. And then in the other districts, we found that it was more evenly distributed like throughout the day. And that obviously has different implications for, I mean, if you're thinking like, okay, can we move those water contacts to different times of the day, right? I mean, depending on how they're distributed, right, you have different opportunities for like moving them. So for example, like in Pakwatch, I guess that a lot of people are going at the riskiest time of the day. So I mean, there you could have probably the biggest impact out of all of the three districts by incentivizing people to go at different times of the day, for example. But I mean, then in some of the other districts, it was kind of more evenly distributed, so like a little bit less opportunity to do so. But I think it's certainly a very interesting thing to consider, especially given that like maybe it's a bit less disruptive than other kinds of behavior change, because people can still do the same things, to just try to reorganize their days to do it at a different time. So I think it's definitely something that needs to be investigated more. And then regarding Anna's question around the intensity, I mean, that's also a good question. And actually on one of the slides, like, well, you may be able to see it in the recording. And also if you look at the paper, is like that actually that for the intensity of infection, like we saw a little bit more of a similar gradient to kind of the water contact, where we did see that when we looked at like the percentage of people who had like heavy infection, that actually did decline somewhat with distance to water bodies. So telling us that, okay, like those heavily infected people, they are also closer to water bodies. But I mean, it's ultimately also a question of like, what do we really care about, right? Do we only care about those people that are heavily infected, or do we care about like everyone? And I think that we should care about everyone as well. But I mean, obviously also given that resources are kind of getting more limited and so on, I mean, that's definitely something that can also be and should be taken into consideration as well.
Thank you very much, Fabian. And before we move off from maybe the topic of the data itself, just a quick point from Godlove Bunda Webninje. Thank you for the brilliant presentation. Just one question. The different activities were reported by the participants, or were they observed carrying out these activities? It's to note that the degree of contact captured by the questionnaire may not actually reflect the ideal exposure or the duration spent in the water.
Mm-hmm. No, that's again a very relevant question, and also like let Juliette chime in if she wants to. But I mean, so these activities were reported generally by the household head, but in the presence normally of all of the different people that lived in the household. We do believe that those activities like reflect like the activities that people were truly doing, and we also investigated that a little bit by kind of comparing the survey responses with also the direct water contact observation that we did, where basically somebody was sitting at the waterside and recorded each and every contact that was happening. And we generally see that the patterns, the overall patterns from the direct observation and from the surveys, they do like match up. So, I mean, we don't see that, oh, there's generally, you know, one age group, say like teenagers, which are like hugely underreported or something. Like we don't see that. Then, of course, for the specific activities, it is a little bit difficult to say that, you know, like all of the activities are necessarily accurately reported. But I think that from the analysis, like we do feel actually somewhat confident that like even these surveys do give us like a good understanding of the different activities that people do. I mean, we do note that like for recreational activities, those are not so well represented among the respondents. So, I mean, we wonder if there's like some underreporting here, but I think that definitely for the other activities, like we do think that they should be like relatively well represented among participants.
Fantastic. Thank you, Fabian. Perhaps turning to Juliette and Narcis. First of all, would you like to add anything to Fabian's thoughts and words, build on that? But maybe opening the lens a little bit more towards the policy or the application in terms of disease prevention, disease control programs. What is the potential of human water contact or this kind of precision mapping exercise to actually identify infection hotspots? I'm not sure if Julia or Narcis would like to.
So, some of the results that weren't presented today, and Fabian can elaborate, we're actually using these models to predict reinfection, and they do quite well. So when you take self-reports or direct observations or other measures of water contact and try to correlate that with infection, they don't correlate very well. And the determinants are quite distinct. And I think that is kind of presented in this Nature comms paper that Anouk has kindly provided the link to. But what was really nice was that you could predict reinfection quite well with these spatial models. So we think that if you're focusing on where people are getting reinfected after mass drug administration or another form of treatment, then it would align very nicely with thinking about where the hotspots would be, especially if hotspots are defined as a non-response to treatment.
And so perhaps this could be a question for you, Narcis, in terms of these mass drug administration programs. And we know that resources, funding fluctuates, comes and goes. So what is the easiest way to incorporate or apply these models and this work to actually improving where resources, where interventions should take place?
Yeah, as I said earlier, our guidelines, the WHO guidelines say that in hotspots, you treat everyone at least twice a year. But remember, the drugs are missing funds to distribute the drugs is inadequate. So this study has shown that it is not necessary actually to treat adults twice because the reinfection is very long, is much lower than the children. So children treat them twice, but adults who are more
Just give Narcis a minute to reconnect.
It shows that when you it shows that with it we're also looking at infection in the synetes and so on. We see that we can from this tell that people who visit the lake or the water site early in the morning or late in the evening, those are less risky water contact points. So that's very important. And another thing, we have been thinking that you go 10 kilometers but actually you concentrate all of these, your services, very close to the water. Because people, that's where they get water contact. And if you put a latrine 5 kilometers, 2 kilometers, and so on, it is not going to assist you in reducing the risk. Just concentrate within 400 kilometers of the water bodies. And that one is a very, very important also section. Yeah.
Thank you. And that ties in really nicely with a question from Emmanuel Marimi. Again, great presentation, SchistoTrack team. Just curious about the high-level policy implications of your recommendations, especially in resource-limited endemic areas. How feasible is the uptake of your recommendation? So now not to look at health, but to build additional boreholes closer to major water sources such as Lake Victoria. And that also follows quite a nice exchange in the chat regarding actually the quality of the water, an issue brought up by Yale Velleman. Using boreholes to reduce water contact, this depends to some degree on whether the boreholes meet user preference criteria, functionality, taste, hardness, yield, and so on. And also drawing on an example of collaboration with the Ministry of Health, Ministry of Water and Engineering. I think in Eastern Uganda, water hardness came up as a major problem for borehole use. Sorry, that was a long-winded way of asking. So not only in terms of looking at health policy implications, but also with completely different departments and WASH interventions. How open are program managers, policymakers to uptaking your conclusions to improve infrastructure? And how can that be improved?
Perhaps we should be careful here in that we're using available information on the usage of existing infrastructure that's commonly built. And we're trying to understand how often that is used. So the points about the hard water and the boreholes are good points. Maybe that's why you can't crowd out more behavior because of the preferences. So whether the technology is going to be another borehole or something else would need that qualitative work, which Fabian alluded to, of what exactly you do build. So we're not recommending take exactly a borehole and build it next to our home. We're just trying to say, if you're going to introduce infrastructure, how does space play a role? And what do we think we can divert based on the usage patterns that we can learn from existing infrastructure, given that's all we have? So I think that's really quite important. So someone had made a comment about the use of external data from satellites like WorldPop for validation. So one of the slides that Fabian had presented actually was expanding the study data to use WorldPop data of household locations. And then you can kind of validate that data if you have a bit of ground truth, because WorldPop estimates what it thinks is a home based on various thresholds. So you do need some real data to know what are households and then think about the extrapolations from that. But what was very nice was that you could use WorldPop data with these models. So they would be easily applied in other areas. And the idea would be just to give you a mapping of where the activity occurs and where the diversion might happen if you were to build infrastructure. So I think that it's kind of a fine line. And if new technologies are available or alternative sources that better suit the preferences of individuals, then those should be pursued. Because we also know that in our study areas, people say the lake water is sweet, and they go and they go to the lake.
Yeah, I think maybe also just one add-on point to this, which I think is important, is that, I mean, obviously we're living in an area where there's more limited resources maybe for implementation for these programs. But I do think that this work is also important because it can basically help us understand, okay, like imagine we just have the money to just build one borehole, right? It can help us to identify like what is the best location for this, like where should we generally build this so that like the resources that we have, that we can use them like in the most effective way. And we even saw that like in a lot of our study areas, there were also a number of boreholes that were like fell into disrepair and that weren't really used, right? I think that obviously there are also a lot of questions around like maintenance or, you know, saying like, okay, like which boreholes should I fix if I only have a limited amount of money to just fix one borehole, right? So I think that for all of those things, like even if you don't have a huge amount of money, like theoretically, I think that our work can help to like see, you know, what's the most effective thing that we could do, you know, even if we don't have like a huge program to build like new taps for boreholes.
Fantastic, thank you. More and more questions in the chat. I think we can't have a webinar about schistosomiasis without talking about snails for a few minutes. So Silvana Bello was asking, did you have data on snails infection? And also very interestingly in the chat, Alphonse Renz suggests, would it be possible to estimate a sacarial density index by combining the numbers of intermediate snails per place, square meters, with the percentage of Cercaria shedding snails? So yes, how do you position the snails in your work?
Maybe, Fabian, you can talk about one result that we didn't present was related to the contaminated sites, which would also relate to Charlie King's comment about the difference between sites that are used by animals or not, or have visible contamination.
Yeah, I mean, this is a really like important question, and like maybe I'll just give 2 pointers to this. I mean, the first one is that even in the systematic review, like we were also interested in like studies that like had these sacarial exposure indices and so on. But I mean, it's really complicated, and like I mean, every study has come up with their own custom way of doing it, so they're really like not very comparable like across studies. So I think that like, if we think of, I think it's a valuable thing, but I feel like we really need to have like more of a better understanding of like what should actually go into it and how should we do it. Because I think that, again, I think it's a bottom-up approach where we say, okay, like let's first understand like the environmental risk well, let's understand water contact well on its own, and then we can think about putting it together, because otherwise it just becomes like very confusing very quickly. Because I mean, even in our study, we also saw that there's a lot of variation in you know, the number of snails at different sites, like the seasonal variation and so on. So that's really where things become very, very complicated to measure. So I think that we should really be working towards it, but also I think like understanding one thing at a time, I feel like is maybe a bit more helpful than trying to capture it all in like a certain way. That's what I would say. And actually, we also had some models even in this Nature Communications paper where we explicitly incorporated also like, what are the number of infected snails at these different water sites that are close to people's households and so on. And we actually found that it didn't improve the ability of the model to predict infection, like incorporating that information, right? Saying that, okay, like it's really complicated, right? Just having those measures doesn't give us that much more information for understanding people's infection status and people's infection intensity just because we know that. But it's certainly, I think, a huge area of research where valuable contributions can be made.
And maybe just a simple point, just to address Charlie's comment, is that the sites that were unused, we could see actually that individuals were less likely to use contaminated sites. The malacologist on our team would record whether there was visible stool at the sites. And then that was one thing we used to characterize the contamination at the sites.
Thank you. And Juliet, you've also added, for everyone who's on the chat, you've added a link to snail data in the content of an article. I also take a moment to thank Anu Guvras and the Global Schistosomiasis Alliance for all the amazing resources that have been posted in the chat as well. But just before we kind of bring this incredible webinar and a huge body of work that you've shared with us, kind of bring it all together. There was here another question by David Rollinson. I'm just wondering whether there's any information of water contamination with urine or feces containing schistosome eggs, or might this occur in more private places?
Well, I think probably that relates to my earlier answer about there is evidence of contamination. We also are collecting and shedding the snails, so we see the snails are infected. We see where infrastructure might have been submerged because there are public latrines near the shoreline, so that would also increase contamination. We also worked on World Sanitation Day last year to provide a map of sites to local district officers when they were doing community sensitization to identify essentially what sites were designated as public toilets and not to go to. So that actually proved to be very useful, is actually just recording what you see at the site. And also the livestock that are moving through the site, we also keep track of that.
Yeah. Fantastic, thank you. Well, we just have a few minutes left. There's never enough time, but maybe just to wrap things up a bit, I wanted to ask the three of you, what are some of the practical ways that you see or you envisage that your model could be validated for policy use?
I don't know. Should I start? I mean, I feel like that one thing that I'm really interested in is that I mean, I think we did a really in-depth study of one context, but I think we need to understand, like, you know, how, to which extent it generalizes around different, like in different areas. So I think that it would be hugely interesting, for example, if there were some, you know, different teams that were doing like very small scale GPS logger studies, like in different countries, maybe with different ecologies, different water bodies and so on, to understand to which extent, like, you know, these models are actually applying. I think that would be hugely valuable to understand this, because I think at the moment we don't know so much, like, okay, to which extent can we actually make general statements about human water contact, and to which extent is it all, like, you know, dependent on, you know, regional factors and cultural factors and so on, right? I think that we really need to develop a better understanding how we should actually think about this. I think that would be hugely valuable in my view.
Narcisse and Juliet, any final words as we conclude our webinar?
I think it would be great to work together. I see some comments in the chat with individuals that are thinking about what to add into mapping exercises when they're, and whether this can help overlap with prevalence mapping exercises and give them more information there on where to treat, given that we're in a severely limited funding landscape. We know that space is important. We have some data that's easy to gather and could be gathered from other contexts. We have the framework and the models set up that have been trained on our very detailed data that hopefully then could be reused to predict elsewhere. So it'd be great to see how informative it could be for other contexts, kind of, you know, building on what Fabian said. So do please reach out.
Yep.
And maybe Narcisse.
Yep. And Narcisse, for our final word of the afternoon.
On my part, hello?
Yep, We can hear you.
Yeah, on my part, I am very amazed. I have been doing schistosomiasis for quite long. I'm one of the people who pushed for treatment of everybody twice in the hotspots in the WHO guidelines. I was voting for that side, but I have seen that really with this study, we do not have to treat adults twice, and that is very, very important to me. I have been thinking that when you are giving alternative water supply and so on, and people who are affected, we've been going up to 5 kilometers, and when we had money, we would go up to 10 kilometers. It is not necessary. Just put the resources down here, and along with the water bodies and so on, and you would receive a lot. And even stationing the water body, the safe water, where you put it, it is not, I thought that you just put water body there, boreholes, and people, you will use health education messages, but people don't want to go there. We have been wondering how we can do snail control or, control on the water bodies. And of course, living very, just isolating it, just a few places here and there, we shall not affect the fish, and yet we shall do monociding if we want, and yet we shall control snails if we want, and we make an impact. So the study has been very important to us, and I will push for policymakers. I will write policy briefs from this and present it to the top level, Minister of Health, and we do we change some things. We can mobilize.
Oh, we just lost you right at the end, Narcis, but that was a fantastic endorsement of this work. And lots, at the same time, lots of compliments coming in through the chat. Juliet Nambatia, thank you for the good presentations. Alan Fenwick, thanks for a fascinating afternoon. Yudia Tuheri, thanks so much for the great presentations, and many, many more. Narcis, Juliet, Fabian, it's been a pleasure having you on this session. It's been an incredible work that you presented. We are really looking forward to keeping in touch about that. And finally, also a big thank you to our co-hosts on this session, the Global Schistosomiasis Alliance, and to our wonderful audience who have been so engaged in the chat. I'm really sorry, there's a few questions that were left outstanding, but I'm sure that Fabian, Narcis, Julia, you're completely open to continuing the discussion either by email or any other means. The SchistoTrack page is up on Oxford's Big Data Institute website. You can also find out more and get in touch that way. And so all that's left for me to do is to wish everyone a wonderful afternoon and rest of the day. Thank you all for tuning in today, and a final big round of applause and thank you to our speakers. It truly has been fascinating, and congratulations on this work. Thank you to all. Until next time.